Welcome to This Explainer. Today, we're diving into the incredibly dynamic, sometimes bizarre, and absolutely fascinating microscopic toolkit that scientists use to bypass a living cell's natural defenses. We're going to look directly at the methods of gene transfer and gene cloning that have literally revolutionized biotechnology as we know it. So let's start with this crucial question, because if you think of a cell as a highly secure fortress, it instantly makes sense why we need such a wild array of specialized methods to actually breach it. I mean, you can't just knock on the door and ask a cell to take in new instructions, right? Cells have evolved over billions of years to keep foreign generic material out. That is exactly how they survive viruses and mutations. But to rewrite the code of life, scientists have had to become master lockpickers. And here's the real kicker. Once we actually get that new DNA inside, the cell itself becomes a biological factory, faithfully cloning those new instructions every single time it divides. Okay, here's our roadmap for this explainer. 1. The gene transfer challenge. 2. Vector-mediated gene transfer. 3. Agrobacterium, nature's plant engineer. 4. Direct delivery, chemicals and electricity. 5. Physical delivery, guns and needles. And finally, 6. Unconventional direct methods. Let's jump right in. Section 1. The Gene Transfer Challenge. Let's categorize our biological lockpicks. The best way to wrap our heads around this is to look at how we approach the challenge. Basically, all of our cloning and transfer methods divide neatly into two distinct camps. On one side, we have nature's careers, the vector-mediated methods. This is where we use biological agents, like bacteria and viruses, to sneak DNA inside. They act like biological Trojan horses. On the flip side, we have the vectorless, or direct entry methods. This is where human engineers step in and use physical or chemical brute force to insert DNA directly, completely cutting out the biological middleman. Section 2. Vector-mediated gene transfer. Sneaking past the guards with nature's careers. Let's check out 3 classic ways nature moves DNA around without sounding the alarm. First up is transformation. Picture this. A bacterial cell just directly swallows up foreign DNA fragments, or a circular piece of DNA called a plasmid, right from its surrounding environment. Just absorbs it. If it works, it either swaps that fragment into its own chromosome, a process we call homologous recombination, or it just maintains the plasmid stably inside. And as this bacterium multiplies, it creates millions of perfect identical clones of that newly acquired DNA. Next, we've got conjugation, which is, well, essentially bacterial mating. A donor cell carrying a fertility factor, the F+ cell, grabs onto a recipient, the F- cell, using this microscopic tube called a conjugation pilus. It literally reels it in, and the donor directly passes a single strand of plasmid DNA right across to the recipient. The recipient then builds the missing half, effectively cloning the genetic instructions and transforming itself into a brand new donor cell. It's a highly efficient, direct biological transfer. And finishing our quick tour of natural vectors is transduction. Now, this one uses a virus that specifically targets bacteria, a bacteriophage, as the courier. But it's actually a brilliant biological accident. During the assembly of new viruses inside a host cell, pieces of the host's bacterial DNA get mistakenly packaged into the viral shell. So when that virus goes on to infect a new recipient cell, it accidentally injects the previous host's DNA instead of viral DNA. Scientists can deliberately hijack this natural mix-up to intentionally deliver customized genetic payloads for cloning. Section 3. Agrobacterium, nature's plant engineer, the original genetic hacker. Now, this is absolutely fascinating. A naturally occurring soil bacterium actually became the very first successful plant transformation system back in 1983. We're talking a massive breakthrough in cloning specific traits into crops. The bacterium is called Agrobacterium tumefaciens. In the wild, it's a pest that causes plant tumors, but to hack it for our own benefit, scientists use a clever 3-step sequence. First, they isolate the bacterium's tumor-inducing, or T plasmid. Second, they gut the disease-causing genes and swap in a desirable foreign gene, like disease resistance, plus a selectable marker. Finally, they just let the bacterium do what it evolved to do, use its special virulence genes to transfer that specific slice of DNA straight into the plant cell's nuclear genome. Once this engineered bacterium infects plant cells growing in a lab culture, the real magic happens. The new DNA integrates directly into the plant's chromosomes. From that single modified cell, scientists can grow an entirely new plant. And because the plant cell meticulously divides and clones its DNA at every single stage of growth, Every leaf stem and root cell in that adult plant is going to carry and express that foreign gene. It is an absolute masterpiece of biological hijacking. Now, while it's an incredibly stable natural carrier for broad-leafed plants, it definitely has its limitations. Its biggest flaw? The host range. It's rarely successful in monocots, which is a huge bummer because that includes many of our most vital global cereal crops, like wheat, rice, and corn. Plus, it really struggles if you're trying to transfer multiple genes at once, and the whole regeneration process from those cultured cells can be agonizingly slow. Section 4. Direct delivery, chemicals, and electricity, when human engineers take the wheel. So, if bacteria and viruses are nature's way, what do we do when we need direct entry without those strict biological limitations? What if we really need to infect a monocot? Well, if a tough plant cell wall is blocking the way, scientists can first strip it completely away to create a naked cell called a protoplast. From there, they can use passive chemical absorption, adding chemicals like polyethylene glycol, or PEG, to alter the membrane's permeability so the cell just passively swallows foreign DNA from the surrounding solution. Or we can use electroporation. By applying high-voltage electrical pulses to the cells and the DNA, we literally blast temporary pores straight through the cell membrane. The DNA rushes in through these holes before the cell naturally heals itself. It's shocking, literally, but incredibly effective for getting genes inside for cloning. Section 5. Physical delivery, guns and needles. The microscopic brute force approach. Look, if chemicals and electricity still aren't direct enough, we enter the realm of purely physical, brute force methods. These act like microscopic construction tools to bypass all biological barriers. First deployed in 1987, this biolistic or gene gun method is exactly what it sounds like, a microscopic shotgun. It uses pressurized helium gas or electrostatic energy to physically shoot DNA straight into target cells. Because of the sheer physical force, it couldn't care less if the plant is a monocot or a dicot. It delivers genes into nearly any desired tissue without even needing to strip away those tough cell walls first. And to really put this firepower into perspective, scientists aren't just shooting a liquid solution. They are actually precipitating DNA onto microscopic gold or tungsten bullets, measuring mere fractions of a micrometer and blasting them right through the cell walls. It completely overrides any natural cellular defenses, which makes it a wildly popular method for those vital crops that stubbornly resist our friend Agrobacterium. On the complete opposite end of the spectrum from a shotgun, we have extreme pinpoint precision, microinjection. In this method, a capillary glass micropipette, which is essentially an unimaginably thin needle, is hooked up to a micromanipulator. The target cell is held perfectly still by gentle suction from a holding pipette, and the DNA solution is manually injected straight into the nucleus of the cell. This bypasses everything and sets the stage perfectly for immediate gene replication and cloning. But as you might guess, such extreme precision comes at a heavy cost. Because a tough plant cell wall would instantly snap that fragile glass needle, scientists mostly have to use bare protoplasts. And because a highly skilled technician has to perform this delicate operation one single cell at a time, it is incredibly demanding manual labor. A top-tier expert might only successfully inject 40 to 50 cells in an entire hour. It's painstaking work. Section 6. Unconventional direct methods. Lipids, pollen, lasers, and fibers. To round out this explainer, we have to look at the borderline sci-fi methods currently being tested at the absolute frontiers of biotech. First up is lipofaction. Instead of shooting or shocking the cell, this method protects the foreign DNA by wrapping it in artificial lipid vesicles called liposomes. Think of them as tiny bubbles of fat. When mixed with protoplasts, these liposomes just merge with the cell membrane via endocytosis. Basically, the cell swallows the bubble whole. Once inside, the cell's own natural enzymes dissolve the fat, safely releasing the genetic payload so it can integrate into the host nucleus and be cloned. It's a brilliant chemical disguise. Or, alternatively, we can literally hijack a plant's natural reproductive system. Scientists wait for a flower to be pollinated. Then, they boldly cut off the stigma, exposing the growing pollen tubes inside, and drop a DNA solution right onto that cut surface. The whole idea is that the DNA diffuses straight down through the germinating pollen tube, directly into the ovule, modifying the zygote at the exact moment of conception. It's incredibly simple in theory, though achieving consistent, stable clones this way is definitely an ongoing challenge. And hey, if that wasn't wild enough, check out these other experimental methods. Macro injection uses actual hypodermic syringes to inject DNA directly into developing floral tillers before they even form reproductive cells. Laser-induced transformation involves literally sapping cells with a laser microbeam to burn tiny transient pores in the membrane for DNA to slip through. And fiber-mediated delivery? That uses silicon carbide fibers just 0.6 micrometers wide to physically pierce the cells in a suspension culture. It acts like thousands of tiny microinjection needles plunging in all at once. From naturally occurring Trojan horses like Agrobacterium to blasting microscopic gold bullets and even zapping cells with lasers, the sheer ingenuity of these gene transfer and cloning methods is just staggering. We've learned to breach the fortress of the cell wall in almost every conceivable way. It really leaves us to wonder, as our biological lockpicking gets faster, more precise, and more reliable, where exactly will genetic engineering take us next? Thank you for joining me for this explainer.